We've talked about the fact that proteins carry out their function by binding, by recognizing something. And then maybe they catalyze, maybe they do something else, but that binding process is really important. And antibodies are an example of a protein that binds, that has no catalytic activity. It doesn't transport anything, but it has a very important function because each antibody has a binding site that recognizes some chemical, that recognizes some structure. And when it recognizes that structure, it latches on, and because it has two sites, it's able to create a network. And that network gets recognized by the body to create an immune response. We've talked about the fact that most proteins are dynamic, that is, that they are constantly in motion, that they're not necessarily fixed in shape or in position. But there are some proteins that are precisely designed to create movement. And examples are muscle, that's shown on the right, and membrane proteins on the left. You know about the movement in muscles. You have them, that your fingers move, your arms move, your head moves from side to side. That's all has to do with the processes that go on in muscles, and those processes are protein-based. There are also membrane proteins that transport things into and out of the cell, and we're going to talk about both of these. But we'll start briefly with muscle. And there are two basic filaments that are in muscle. They are myosin and actin, and they work together to form the muscle and allow it to move. Muscle consists of these long fibrils called myofibrils, and when they are broken down, you can see that they are together in clumps, and when you come to the bottom, you can see this single muscle fibril, and you can see the myosin thick filaments and the actin thin filaments, and then there are bands that separate segments of these filaments, and it is the movement through that H zone that allows the constriction of a muscle and then, of course, the release of the muscle. And we're going to look briefly at this YouTube to show you how muscle movement is generated.
Myosin binding sites on actin are exposed, allowing interaction of the myosin cross bridges with actin. Release of the myosin cross bridge from actin requires binding of ATP. After ATP binds, it is split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to cock the cross bridge in preparation for the power stroke. The power stroke is initiated when the myosin cross bridge binds to actin. The cycle begins anew when another ATP binds to myosin. At rest, most cross bridges are in the cocked configuration, prepared to interact with actin when the tropomyosin troponin complex is moved away from myosin binding sites on actin by calcium ions.
So a couple of reflection questions. How can proteins whose only function is to bind be protective, like antibodies? And what other types of activities might be generated? What other functions could be generated by just binding, just recognition? And think about why the ability to move proteins with respect to one another is so critical for muscle function. Can you think of another way that proteins might serve as muscles and without the particular design that actin and myosin have? And also remember why ATP is important in this process. Think back to our delta G discussion.